Right now, inside your cells, there is an operating system running on a microscopic 3 billion letter genetic code. It operates entirely on just 4 chemical letters: adenine, thymine, cytosine, and guanine, A, T, C, and G. Every physical trait you possess is spelled out using only those 4 blocks. For most of human history, we were completely blind to this instruction manual. We knew traits were passed down, but the actual machinery doing the work was invisible. That changed in 1952. Using specialized X-ray diffraction cameras like this one, scientists finally managed to capture the physical shadow of the double helix. Seeing the physical structure meant we could finally stop guessing. It was our first real step toward reading our own biological hardware. The physical architecture is a model of efficiency. DNA is a rigid molecule wrapped into a double helix, held together by a sturdy sugar phosphate backbone. This animation shows the strict rules of that structure. The system relies on flawless chemical pairing. Adenine always locks perfectly with thymine, and cytosine always pairs with guanine. When your body needs to grow or repair a cut, that structure cleaves straight down the middle. Floating ambient molecules latch onto the exposed halves to create 2 identical carbon copies. Biologists call this semi-conservative replication. It is resilient against alkaline damage and structural decay, ensuring the sequence remains intact for long-term storage. But DNA is strictly an archive. It sits locked safely inside the nucleus of your cells, and a locked vault cannot build a building on its own. So if the master blueprint never actually leaves the vault, how does the body build bone, muscle, and tissue? This chart highlights the difference between the archive and the active workforce. RNA is a single-stranded molecule operating outside the nucleus. It runs an assembly line. Messenger RNA copies recipes. Ribosomal RNA reads instructions, and transfer RNA fetches amino acids. The body can even halt this entire process on demand. It deploys tiny molecular assassins known as microRNAs. These short sequences hunt down specific messenger strands and cleanly cut them in half before the protein can be built. Once scientists mapped out this active moving pipeline, they realized something crucial. The genetic code is an ongoing, interceptible process. This realization shifted the focus of biology. We moved from simply watching the process to actively steering it. By borrowing a defense mechanism from bacteria, scientists developed CRISPR. It allows us to target and excise specific DNA segments with pinpoint accuracy. This diagram illustrates recombinant DNA. Since all Earth life shares the same chemical alphabet, we can stitch sequences from plants and humans into hybrid chains. Using next-generation sequencing, machines can organize billions of these bases in hours. We have the tools to read the sequences, the chemical precision to edit them, and the ability to test the results in real time. We are now actively programming biological systems. Since 1952, every edit and discovery has operated within the strict limits of adenine, thymine, cytosine, and guanine. But why should we restrict ourselves to the 4 letters nature provided? Look at this model of Hachimoji DNA. Researchers mathematically expanded the standard code to create an 8-letter molecular system. They engineered 4 entirely new synthetic chemical bases from scratch. These new man-made letters, labeled S, B, P, and Z, perfectly integrate with the original Earth bases, binding together to form an artificial double helix that maintains its integrity as effectively as the natural version. This synthetic system performs exactly like the real thing. It chemically meets every structural requirement needed to store, transmit, and evolve information inside living cells. You can place this code into an abstract synthetic cell and watch as it actively divides and evolves entirely under its own power. Hachimoji proves that life doesn't have to be written the way evolution designed it. Now that we have the capability to author the code from scratch, we have to decide where to stop.